Seminars, and today we have truly a very special speaker, friend from many years, and he is going to enlighten us with his most recent research. But before we go, let's Hernan introduce our speaker.
Hello and good morning. My name is Hernan Gomez, and I am a third-year graduate student under Dr. Palczewski here at the Center for Translational Vision Research at UCI. It is my pleasure to introduce today's distinguished speaker, Dr. Simon W. John. Dr. John received his PhD from McGill University in Montreal, Canada, in biology and human genetics. Following this, he was a postdoctoral fellow at the University of North Carolina at Chapel Hill studying genetics and hypertension. Since then, he has had multiple appointments, currently as Professor of Ophthalmic Sciences and member of the Zuckerberg Institute, Columbia University in New York, adjunct professor at the Jackson University at Bar Harbor, and adjunct professor at the genetics program at Tufts University Graduate School. Having contributed to over 140 publications, he has also been the recipient of many awards, including the Sanford and Susan Greenberg Visionary Prize to End Blindness, twice received the Louis Rudin Glaucoma Prize from the New York Academy of Medicine and the Bresler Vision Science Award. Dr. John's research determines molecular mechanisms of complex eye diseases, especially glaucoma, focusing on IOP elevation, ocular drainage, structure, biology and physiology, and neural degeneration. His group was the first to uncouple soma and axon degeneration pathways in glaucoma, including recently implicated perturbed NAD glucose and pyruvate metabolism in glaucoma, followed by significant protection using vitamin B3 and NAD increasing gene therapy. Other notable scientific endeavors, including pioneering the use of mice to study glaucoma, introducing key tools, models, and mechanisms, and discovering that radiation treatments protect against glaucoma and implicates a subclass of monocytes in early pathogenesis. His aforementioned research is supported by multiple R01 grants and additional support from the Bright Focus Foundation and the Glaucoma Foundation. Now, please join me in providing a warm welcome to Dr. Simon W. John for his talk, Towards Resilience, Boosting Strategies Against IOP Elevation and Glaucoma.
Thank you for having me today. It's a pleasure to be speaking here, and thank you for the generous and kind introduction. Today, I'm going to tell you about some of our work that is built to the idea of using resilience boosting strategies against IOP elevation and glaucoma, and possibly other ocular diseases as well. I'm going to give you a brief introduction and then talk about the work that is built up to this. I'm going to be giving the introduction now. The strategy we're really thinking of as being a key change to traditional strategies is to try to boost the resilience of the cells that are undergoing stresses either due to aging or disease. If you can boost their resilience, they can fend off these stresses and disease better, function better, and survive better, and therefore alleviate the disease. The strategies we've been developing are essentially around restoring and enhancing a healthy metabolism. Such treatments, which can be dietary, they can even be nutrient-based or lifestyle changes, are generally gene agnostic. They can work in wide swaths of the population, especially as they'll often address aging changes. But they can also be used against specific genes and specific pathways. And so they complement very much the precision medicine, highly tailored approach. Common features in aging and ocular disease are perturbed NAD metabolism. This has become known from our work and other groups over the past decade or so. You can have decreased nicotinamide adenine dinucleotide, a key cofactor for many enzyme reactions, very important in redox metabolism, mitochondrial health, and energy homeostasis. And there's decreased NAD with aging in different parts of the body and in the eye. And as I'll show you later, this is contributing to glaucoma. Associated with the disturbances in metabolism, we find mitochondrial dysfunction. Mitochondrial dysfunction is not just secondary in some of these diseases, but it's a primary driver. This is also associated with oxidative stress. And of course, you get increased inflammatory signaling with aging. So these resilience factors can address these processes. There's growing evidence from our work in glaucoma and other groups which have confirmed or extended our work and in AMD of increased reactive oxygen species, increased PARP activity, poly-ADP ribose polymerase. If this is activated by oxidative stress and DNA damage, to repair DNA, it can consume a lot of ATP, resulting in energetic collapse. I already mentioned decreased NAD+. There's also evidence for alterations in sirtuins and PGC alpha-1, which is very important in mitochondrial metabolism and stress, some stress responses, and for the perturbed mitochondria. So the treatments I'm talking about, even though we've only tested them in glaucoma rigorously to date in my lab, other labs are working on other diseases, and we are planning some trials in AMD as well. So let's come to glaucoma. Glaucoma is a major cause of blindness, affecting about 80 million people worldwide, but in another 20 years, it'll be over 100 million people. They are a complex group of diseases that result in death of retinal ganglion cells and degeneration of the optic nerve. To show you glaucoma in a mice, here you can see a normal, healthy optic nerve head. This is the central retinal blood supply. This is the nerve fiber layer where the axons of the retinal ganglion cells accumulate, and it's thickened by the optic nerve head. If you look in glaucoma, this nerve fiber layer is lost as the axons are lost, and you get atrophic remodeling or excavation of the optic nerve head. And in most glaucomas, it's primarily the retinal ganglion cells, the inner nuclei of cells that's damaged. You can see there's many nuclei missing here compared to the control, but the rest of the retina is largely intact. Primary risk factors for glaucoma are genetics, family history. Age is one of the major risk factors. And intraocular pressure is also a major risk factor, and it's the only one that's been traditionally modifiable. And so all approved current treatments aim to lower intraocular pressure to a safe level. Because you need to understand this near the end of my talk, I also want to introduce the ocular drainage structures. Much of our recent work has been in the anterior segment that I'm not speaking about in any detail today. The aqueous humor is made by the ciliary body. It then flows to the posterior chamber through the pupil and into the angle, this angle made by the iris and cornea, or the iridiocorneal angle. And in this angle, the drainage structures are located. And if we look on this left-hand side, a blown-up angle, this is the iris, you see the angle. There's an elaborate resistor with different specializations in different parts of it, known as the trabecular meshwork. The aqueous humor has to percolate through this trabecular meshwork, and then it has to enter this Schlemm's canal. And this endothelium of Schlemm's canal is the final barrier to aqueous humor exit from the eye. And a major site of resistance is in this region of the inner wall of Schlemm's canal and the juxtacanalicular trabecular meshwork. Now, despite many, many studies, neither the biology of these tissues, exact molecular classification of the cell types, nor mechanisms underlying IOP elevation in most glaucoma are adequately understood. So my group's been one of the groups trying to do a lot of work to change this and advance the field. My group keeps a grounding in genetics for most experiments, but also heavily in genomics. It's a very powerful set of tools, as you see with the survey here, for understanding disease mechanisms. You don't need to worry about the details on its slide. I just wanted you to understand we comprehensively use basically all genetic and similar types of tools. In recent years, in addition to understanding mechanistic biology at a deeper level as we can, we've had to do a lot of tool development, because in studying the anterior chamber, the basic tools haven't really existed adequately for mice or other species in adequate ways. So we've developed different methodologies, including relatively simple ones such as flat mount traps, methods to isolate Schlemm's canal and trabecular meshwork cells, improving on what's been in the literature, perfusion systems to study the physiology, etc., etc. And we're currently initiating a large effort on viral variants to specifically target the Schlemm's canal and trabecular meshwork. We're using viral evolution types of strategies and we hope to get subtype specificity. You can see some initial images over here with some viruses that infect the trabecular meshwork with a cherry. The Schlemm's canal was in a green fluorescent body mouse that was already labeled. Or we can, on the contrary, with other viruses, infect Schlemm's canal but not the trabecular meshwork. We've also been developing panels of mutants to lead us to biomechanistic biology. And so we've developed a unique panel of new mutants. So we have a lot of tools now, but they still need improving. In addition to tools, we've really had to do basic characterization of the cell types. So in a paper just published on eLife, we did a detailed characterization of the Schlemm's canal endothelial cells. In this paper, we defined 3 transcriptional states of these endothelial cells. We haven't called them subtypes because they're quite close to each other, and we don't yet know if they always have these different states or they just respond into environment. We've also shown that Schlemm's canal cells are molecularly between vascular endothelial cells and lymphatic endothelial cells, but their state is closer to lymphatic than vascular. There's some errors in the literature on that point, and our latest paper corrects some of that. Here you see some UMAP representations, some single-cell-seq data, and here you see these different classes of inner wall 1 cells, inner wall 2 cells of Schlemm's canal, the outer wall cells, lymphatic cells, blood vessel cells, etc. We've done similar studies, and we're writing up some of this work now in the trabecular meshwork, and we've defined 3 very robust subtypes in this case, which seem to have compartmentalized functions, but we haven't mechanistically or functionally done the experiments to prove that yet, but that's how it looks. We've also done a lot of work on the drainage structure development in the eye. The drainage structures are critical in regulating intraocular pressure, this trabecular meshwork and Schlemm's canal. My group did a lot of work in working out the developmental sequence and showing there's novel developmental processes happening here and vascular development for Schlemm's canal. These structures are not only important for adult glaucoma, but they're very important for childhood glaucomas. And childhood glaucomas are notoriously difficult to treat. So we're hoping by studying these structures, we can especially make a difference in childhood glaucomas and the paradigm of treatment there. But this work is also relevant to adult. Here you see a time sequence analysis from early development. A lot of these structures develop postnatally, so it's postnatal day 2.5 out to P90. We've gone, I'm only showing you P60 here with the trajectory of development. So we're learning a lot of molecular information. This has been in collaboration with Dan Stamer and others who I know was on the talk. We're very excited by the notion of dietary interventions when I come later in the talk to alleviate disease in these developmental glaucomas. In these developmental glaucomas, young infants within the first year or 2 of life will have multiple surgeries, and it isn't always successful, and they need repeat surgeries, and they can lose substantial vision and have a lot of pain and photophobia. The current state of treatment is not adequate, and we're hoping that we can complement it with new dietary types of inventions that are hopefully relatively safe and easy to administer in children, and would hopefully be safe and tolerable over the lifespan of those children. So we're hoping this work will contribute to a new paradigm in the treatment of this form of glaucoma. To show you one model introduction quickly, I mentioned we've developed the NU mutations. Well, we developed an allelic series in the LMX1B gene. We identified these by phenotyping, but indeed we had 7 mutations in the same gene, showing it's important in mice, but it's also very important in human glaucoma. And mutations in the human gene cause high intraocular pressure, and they contribute to both childhood and adult onset POG, or primary open-angle glaucoma, the most common form of glaucoma. LMX1B encodes a limb domain transcription factor. We've got this 7 allelic series affecting different domains. We've been characterizing this allelic series, and what our current data suggests is mutations in the homeo domain. If they make a functional protein that's damaging, these mutations are more severe and earlier onset, whereas the milder mutations in other parts of this protein, at least some of which are nulls, as we're working it out, have a milder later onset form, so resembling the human disease in the spectrum of phenotypes. I'm going to show you at the end of the talk, we've now got data not completely proven yet, but very strong data in this early onset V265D mutation for early mitochondrial stress and abnormal stress responses contributing to TM cell death. And in that, and in this later onset model, which has some similar mechanisms, we're now starting to apply resilience-based therapies. So that's why I'm introducing this. So let's go to the history of these studies. We first came to metabolism and resilience by studying neurodegeneration in glaucoma, the death of retinal ganglion cells in the retina. We uncovered through microarrays initially in collaboration with Abe Clark, who I noticed is on the call. Abe was very instrumental in my early career. Thank you, Abe. And also congratulations on the Barony Prize, well deserved. So anyway, in these early studies, we started understanding molecular processes that were changing. When we came to more sophisticated single cell type studies or bulk sequencing of retinal ganglion cells, we uncovered very, very early metabolic stress and metabolic perturbations. And we've gone on to show that these contribute to age and disease and this neurodegeneration in the mice. Our current data in the LMX1B model is also supporting a role for similar pathways in IOP elevation. So I'll go through those two stories with you quickly. And then if there's time, I'll mention some clinical trials that are currently underway. So the neurobiology of glaucoma using DBA2J mice, an age-related inherited model of glaucoma. It's a form of pigmentary glaucoma. It's a severe form of pigmentary glaucoma with similar etiologic pathways underlying the onset of the pigment dispersion as occurring at least a subset of human pigmentary glaucomas. This pigment dispersing disease disperses pigment and debris into the drainage structures, which induces a damaging response that elevates the pressure and subsequently leads to glaucoma. In these mice, there are two key mutations. We've shown there's about 7 loci or more in all contributing to this, but the two main ones are GPNB, which is a stop codon mutation, and tyrosinase-related protein 1. Both of these affect melanosomal pathways. They affect the iris, but the iris cells under stress and they release pigment. This leads to increased IOP over these timeframes. The iris disease is really getting full on by 5 to 6 months. By 6 to 8 months, you're starting to get increased IOP. And by 10 to 12 months, you're developing the glaucoma. I mentioned these genes because I want you to remember GPN and B. We were able to recover, this is a spontaneous mutation in DBA2J that our research showed occurred somewhere in the early 1980s. We were able to recover a strain of mice from 1982, which had the wild type allele. It's exactly the same DBA2J mice, but wild type for this mutation. We backcrossed that mutation into our modern day colony to make sure it was clean. So we now have a control strain that only differs in a known point mutation that's genetically matched to all of the other DBA2J mice. And alone, it does not, within having this wild type gene, the DBA2J mice do not develop elevated IOP and glaucoma. So it's a wonderful control strain. That control strain is beyond all of the later studies I'll show you, but I won't always be mentioning it. So what are the early molecular changes happening in glaucoma? You've got the cell bodies in the retina, you've got cells in the optic nerve head as well. So there's lots of cell types that could be involved. We've been targeting different cell types for our studies. In some studies, we've just studied the retinal ganglion cells, in others, the optic nerve head, for example. We've used genome-wide expression studies, which we specifically designed to allow us to identify very early stages of disease that precede neurodegeneration. So we developed some methodologies for this. And I can't go through them in detail, but I'll quickly show you the concept. We then progressed to cell type specific studies using these same methods, so at retinal ganglion cells. So we could ask what increases neuronal vulnerability in glaucoma and what increases vulnerability to IOP with age. This is a slide to quickly introduce you to the concept. Basically, in one of our early studies, we could take the optic nerve head and we'd take it from 10.5 months of age. A key age in IOP has been high for a number of months, as per the previous slide I showed you. But many of the mice do not yet have glaucoma by any conventional measures of the disease. It's just high intraocular pressure. So at this age, we can identify some mice with moderate glaucoma, some with severe glaucoma by studying their optic nerves. But most of the mice do not have glaucoma by any criteria conventionally. And so we can't distinguish these eyes to each other, but they've had a higher IOP for months, some of them. So we reasoned some of these mice are going to be at early molecular stages of glaucoma prior to other changes. And that's why we wanted to stratify these mice to understand early stages. We also do a GPNB control mice, which could be housed in the same cages, age, sex, strain, environment matched. And so these were our controls. So we sequenced a lot of tissues, or microarrays in our first study I'm showing you here with Abe. And we were able to then use hierarchical clustering. We put all of these samples into the computer unidentified, unsupervised. We could use hierarchical clustering then to group these samples based on their molecular similarity across the genome. And this allowed us to group them into substages, and we could see if it worked. What we found in an unsupervised fashion is we recreated the GPNB control group. We recreated the moderate and severe stages of glaucoma, and we substratified these mice that we previously could not distinguish into having different stages of glaucoma. And if you look at principal component analyses or other methods, these are clearly distinct. I don't have time to show you that, but we have validated this stringently. And if you go across these groups, they're in this order, you get increasing degrees of differential gene expression in relation to control. So more differentially expressed genes in this direction, so show an increase in disease. I'm not going to show you the results of any of those studies. I want to come to the retinal ganglion cells. For the retinal ganglion cells, we used similar approaches, but in this case, we FACS purified the retinal ganglion cells. We used a dual study design in this case. We had our control, which doesn't develop IOP or glaucoma, age matched to mice at an early stage where they've had ongoing IOP elevation, but they do not have glaucoma by any conventional means. So these are age, strain, sex, environment matched. So any molecular differences here, with all assumptions are correct, they do early stages of glaucoma, and we have a way of controlling for genotype effects due to this gene. We could also age just the wild type mice. And so here we could look age dependent effects. And so we can ask what's due to age or what's due to glaucoma. When we did this for retinal ganglion cells and we focused on the early stages, we identified 4 groups of mice. And as I'll show you in a moment, this group 1 is the earliest. It's indistinguishable to control by any means, even though they're DBA2J mice. And some of these mice get glaucoma, or most of them do ultimately. But these mice haven't yet had a high enough IOP or enough IOP exposure to make them change from control. These groups are then distinguishable from control on the earliest stages where we could detect as having active molecular stages of glaucoma. Let me show you that. So it's this study design I just showed you. We sequenced 82 samples, about 25,000 cells in each sample by bulk RNA-seq. And if you look at this dendrogram over here, here you can see 4 months before IOP, 9 months when mice might have had a high IOP for up to 3 months if they're the DBA2J mice. But the control mice, GPNB plus, do not ever get high IOP. So our controls and our young DBA2J mice all segregate with this DBA2 group 1. That's why we say it's not distinct from control. And then we get these 3 other groups, 2, 3, and 4, an increase in order, which have molecular further distance from the controls. And you see that plotted here in terms of the differential gene expression. These colors only refer to this plot, not over here where they're all green. So groups 2, 3, and 4. So group 2 is the earliest stage of glaucoma we can detect. Group 4 is still very early. It's prior to any neurodegeneration, but it looks much more severe. It's definitely more severe than these other 2 stages. This data implicated mitochondrial dysfunction on metabolic stress in glaucoma. I need to go through some of this quickly. Here you see from these groups of mice I just explained here about control, and then these progressively early stages of glaucoma. If you look at mitochondria to nuclear read ratios, they're perturbed early, suggesting mitochondrial dysfunction. If you look at various pathways, EIF2 important in metabolism, mitochondrial biology, mitochondrial dysfunction, metabolic regulators, and OXPHOS key in energy metabolism, they're all changed at this very earliest group 2 of glaucoma that we can detect. To zoom in on OXPHOS, G is GPNB controls, then these 4 stages. Blue is a lower gene expression, red is a higher. Each little bar is a single gene. These are all of the genes in the OXPHOS complexes. What you can see, group 1 doesn't look very different to the control. Groups 2, 3, and 4 look very different. Supporting changes to this pathway and energy metabolism. If we do EM on mitochondria, very limited images I'm showing you, but they lack cristae, and you see the histograms here. So there's evidence of mitochondrial changes very, very early, and so likely a primary driver. Finding this, we decided to do metabolic studies. We did both targeted and untargeted metabolomics to look at this. And in some of the targeted assays, we uncovered reduced levels of NAD and of glutathione, a key antioxidant. This is consistent with altered energy metabolism susceptibility to oxidative stress. Look here at the optic nerve with increase in age in mice that develop glaucoma, and NAD is going down with age. And remember, the mice are starting to develop glaucoma between these two windows. Over here, if you look at just GPNB control mice, so they do not develop high ion beam glaucoma, in the retina, we still see decline in NAD. So this decline of NAD is an age-dependent effect, but it can get exacerbated by disease. And here you see glutathione. So this led us to the hypothesis that an age-dependent decline of NAD may be key in neuronal vulnerability in glaucoma, increasing the probability of metabolic failure when retinal ganglion cells are exposed to ongoing intraocular stress. We know our control mice have this reduction in NAD, but they do not develop glaucoma. So the reduction in NAD alone is not sufficient. It's too mild. But when you add IOP on top of it, our hypothesis was this allows glaucoma. To test this, and if this is one of the age-dependent susceptibility factors, if you increase NAD, you should lower the likelihood of neuronal failure and degeneration. We decided to do this after thinking hard about it and through discussions with my mentor, Oliver Smithies, a wonderful scientist, Nobel laureate, who sadly passed away a few years ago. But Oliver was involved in this study, which was pleasing for me. So anyway, we decided to use nicotinamide vitamin B3. This nicotinamide form of vitamin B3 goes through the salvage pathway from NAMT to NMN, nicotinamide mononucleotide. And then NMN is converted by nicotinamide mononucleotide adenylate transferase into NAD, an efficient and major pathway forming NAD in the body. So we decided if we give nicotinamide, which is known to be relatively safe, overly available, this might become a real intervention through nutritional means. So we were excited to test this, and so we tested it. Indeed, we found that dietary administration of nicotinamide profoundly protects from glaucoma in this DBA2J model. It increased NAD levels when you feed it. I'm not showing you that. It prevents the loss of pattern electroretinogram. Here's a 6-month mouse with a normal retinogram. Here's a 12-month DBA2J mouse where it's lost. This is a measure of ganglion cell activity. And here's a nicotinamide-treated protected mouse. If you use cholera toxin, putting it into the eye, into the vitreous chamber, so it'll get transported to the optic nerve and back to the brain. In our control mice, it's transported beautifully and it makes it to the brain. In untreated DBA2J mice, it gets blocked at the lamina region of the optic nerve head, similar to what happens in human glaucoma. Axonal transport gets blocked there. It does not make it to the LGN or the supraclaviculus. If we treat the mice, this is our lower dose treatment. It's now transported all the way back and makes it into the brain. So we restore axon transport. Actually, we've restored every measure that we've assessed, including synapses in a wild-S model I'll tell you about in a minute. If we look at mitochondrial morphology, here's control, here's D2 of these abnormal cristae, and here's the treated. So really, it's a very protective treatment. We also protect retinal ganglion cells and axons, very important to even couple these processes in the disease. If we look at retinal ganglion cells stained with RPBMS, here's a control retina of affected glaucoma and a nicotinamide treated protected. On the optic nerve, had a beautiful optic nerve, a glial scar, a damaged optic nerve. Let me explain this plot over here. This is very important because it's giving you an idea of numbers of mice, not just showing you one or two. This glaucoma is highly variable at any age. As you can see here in an untreated mouse, at this 10.5 month time point. No, this is a 12-month time point, sorry. And here you see mice shown in green. I'm calling them no early. They're the mice I showed you in the earlier slide. They have no detectable signs of glaucoma. They appear completely normal. but they might have early molecular stages. So that's why we call them no early, because we can't distinguish. We have mice with moderate glaucoma, typically about 30% axon loss, and severe glaucoma, greater than 50% axon loss. If you just want to look at the no earliest, the no glaucoma, essentially, to make it easy, you can see as we start nicotinamide either early or late, we get very strong protection. Actually, our high dose of NAM decreased the risk of developing glaucoma by tenfold. This is a really remarkable result, but it's highly robust. We've confirmed it repeatedly, and other labs also having robust effects using these treatments now. Now, not only did it stop the glaucoma, it also stopped age-related changes. And so I'll show you some of these molecular protections now. If you want to look just molecularly in the glaucoma, remember this complex, the electron transport chain I showed you, use our GPN and B control, use our 4 stages of glaucoma. Look at this nicotinamide treated. It looks essentially identical to this no glaucoma control. So this is the disease process. If you look at all of the genes compressed in the genome, when you look at these heat maps, excuse me, these are the controls. These are some mice with stage 4 glaucoma, and here's the nicotinamide treated. Look at this little white bar, pale, which is gone in the control, but present in the treated. Look, this is all red. It's blue in the glaucoma. It's red again in the treated. This blue bar down here in the control, gone in the glaucoma, back in the treated. It's really a pretty remarkable protection. If you look at specific pathways by RNA-seq, if you look at the group 4 DBA2J on these different pathways, say oxidative phosphorylation as an example, any gray dot is a non-significantly changed gene between the two groups, the treated and the untreated. A red is a significant different gene between DBA2J and in this case group 4. Here's the nicotinamide treated versus the control, and you see there's no significant different genes. And this was remarkably so across these pathways. When we looked at age-related changes over this 4 to 9 month time point, there were about 4,500 genes normally changing, but only 2% of them changed after the treatment. So it also seems to be retarding molecular aging in the eye. What's the safety of our studies? I don't have time to go through this in great depth, but there's been lots of studies with high doses of nicotinamide in humans for years with a relatively safe nicotinamide or nicotinic acid with a relatively safe safety profile. There are rare instances of liver damage. 3 grams a day, which is equivalent to the lower dose that we used in our studies, when you do the species conversion, is considered safe. We have had one adverse event in a clinical trial which reversed in going off it, but it seems to be a rare event. So you have to monitor patients, but overall it's largely safe, but you should take caution. So our low dose was actually equivalent to 2.57 grams a day for a 60 kilogram person. Our high dose, which caused that very massive protection, was equivalent to about 10 grams, which suggests that by itself, there's probably too much nicotinamide to be given people. And so we have to rethink that. There was a recent study implicating vitamin B3 in vascular inflammation and severe cardiovascular disease. Already for substantial cardiovascular disease, but that's something we'll have to watch out for. So what about other NAD increasing therapies? And can we learn more about the mechanism? Well, we used the wild S fusion gene. This is a fusion gene encoding NMNAT, one of the enzymes in that NAD salvage pathway. It's a spontaneous mutation, but we looked at it in the glaucoma. We've also done gene therapy using NMNAT directly to assess it as a potential one-time treatment in a very complex disease. And it allowed us to increase the enzyme through the gene therapy and the substrate through diet to look at the mass action effects. Importantly, both of these experiments only share the NAD mechanism with nicotinamide. Nicotinamide has other effects in modulating calcium signaling, antagonizing endothelins and vasoconstriction, promoting blood flow, promoting autophagy and mitophagy, inhibiting PARP action, and other things, all processes which we believe may be important in glaucoma. So one of the reasons this may work so well is these pleiotropic effects. But anyway, we wanted to ask and try to get a better idea of the role of NAD intracellularly itself. So using wild-S, we were able to increase NAD levels, and it was profoundly protective from glaucoma. Again, if you only want to look at the no glaucoma, the green, to make it simple, but I'm showing you the whole distributions. What I should have mentioned on the previous slide is in these experiments, we're using sample sizes typically of at least 60 and often over 100 in any group you're seeing to make sure they're robust results given the variability of the disease. You can't use sample sizes of 10 or 20 and get robust results in this model, unless it's complete protection all of the time, which is unlikely. So looking at these mice with no glaucoma in green, you see you give wild acid increases, you give wild acid and nicotinamide, and now 94% of the eyes did not develop glaucoma. Pretty remarkable result. When we looked at these mice and we did Scholl analysis of synapses, etc., we restored the dendritic complexity. The NMNAT gene itself, we added it with an AAV2 intravitreally. Again, NMNAT is this stage. We transduced about 80% of the retinal ganglion cells, and this is a successful gene therapy in this complex disease. You can see the untreated, the NMNAT, or again, the NMNAT with nicotinamide, giving us a strong effect, and we quantified all of this and did physiology, and indeed, it's a strong effect. So those two experiments very strongly support a major role for NAD itself within the retinal ganglion cells, though further studies are needed to fully resolve that and know if there's also roles in other cell types. So do other metabolic disturbances contribute to glaucoma? Well, RNA-seq studies, again, we've just identified alterations of glucose and pyruvate metabolism at the RNA level, as you see here. Again, non-differentially expressed dots, not significantly changed, are gray, significant change to controls are red. And you see across these 4 glaucoma stages I told you about, you get pyruvate metabolism and glycolysis defects. So our data overall supports disturbances of glycolysis. Pyruvate transporters, carboxylate transporters, and other genes also change. So we wanted to ask, does decreased glycolysis and pyruvate contribute to glaucoma? And if so, if we treat with glaucoma, we should have an effect. So glycolysis is the major source of pyruvate. Metabolic studies have shown a massively increased glucose level in the DBA2J retina, but doesn't seem to be systemic, so it seems to be a retinal effect. So our data support this deficiency of glycolysis, and other labs have been working on some of the metabolic pathways as well. So if we go through glycolysis to produce pyruvate, and then this can also generate ATP through these other pathways. So a reason that supplementing pyruvate could overcome any defects in glycolysis, as well as supporting energy metabolism through these pathways. So this is what we tried. The other point I want to point out is pyruvate makes acetyl-CoA, xylacetoacetate can make citrate. Acetyl-CoA and xyloacetate, make citrate. Lower citrate levels have been reported in human glaucoma. So this is interesting. So we did the experiment in mice, and indeed we found another robust effect of pyruvate. If you look at this, you can see pyruvate treatment protects in the glaucoma, not quite as well as the NAM alone. And when you combine them, it's more significant. Or if you look over here, just the mice developing glaucoma, as you give them multiple combination treatments, the combination is the best treatment. Pyruvate has a number of functions in addition to its role in energy metabolism. It's a direct ROS scavenger. It induces antioxidant defense. It can promote autophagy. And so we believe from these results and these combined effects, a combined treatment of nicotinamide and pyruvate will ultimately be the most effective. And coming back to that very high dose of NAM that we wouldn't use in patients, using combined doses with multiple molecules will give us safer treatment profiles through combination. And so that's really what we're working towards a lab. So let me get off the neurodegeneration and come to intraocular pressure elevation. What evidence do we have for metabolic changes in intraocular pressure elevation, and how did we get there? Well, again, even though there's some hints of this in the literature, it's never been clear if it's primary or secondary in various studies. Paloma, who's on the talk, had a nice paper showing disturbances in autophagy in glaucoma. Our current evidence from a lot of gene expression suggests that LMX1B may be regular in mitochondria and possibly autophagy. leading to disturbances and glaucoma. I can't show you all of this data, so very quickly. If we look at our LMX1B mutants I introduced to you earlier on, if you look at the early onset homeodyne V265D mutation, we see all of these pathways changing, suggesting changes in the proteasome and ER, maybe ER stress, but very importantly, our best suggestions now are that mitochondrial changes are some of the very earliest, and LMX1B is known to modulate mitochondria in the brain. So we've got evidence for early mitochondrial dysfunction. We can detect perturbed cell stress responses by single cell sequencing. None of this is published. It's not all ironclad yet, so this could change. But we believe in this V265D model that this is correct at this point in time. We've seen increased trabecular meshwork cell death, so this resistor in the eye, and we see drainage tissue maldevelopment. So all of this is supporting metabolic roles in the demise of the trabecular meshwork and a progressive cell death, only very rapidly in this early model. We believe similar pathways are happening in this late non-homeodomain model because the initial data is agreeing, but this is the weakest data so far. We've really got to study this more and boost it. So continuing on, we've tested nicotinamide and pyruvate treatments. I can only show you a part of the data we've generated, but what I can tell you is given the nicotinamide and pyruvate is proven very promising, and we're very excited by the potential for treating developmental and childhood glaucomas, but also adult onset glaucomas. So what's the data? Well, one you're probably very interested in is we can change intraocular pressure. If we take this early onset V265D model, which is dominant, if we have untreated mutant mice, you see many of them are getting high IOP at an early age. If we treat them with nicotinamide, we have quite a strong effect on IOP. If we treat them with pyruvate, we also have an effect, though it's not as strong. And if we do the combined, we have the best effect on IOP if you look at the box plot here. So the combined treatment seems the best, though we're boosting numbers and boosted ages. Along with this IOP elevation, we see increases in apoptosis, or maybe not apoptosis, I should say cell death at this point in time. If you look at untreated mice, the wild-type mice, we don't see cell death. We've shown this extensively in mice previously when the angle develops. In the V265D mutants, we see increased cell death, and the combined treatment really knocks it down. Now, what does that do for the development of the drainage tissues? Well, in red here, you see in Schlemm's canal, in an untreated mouse and in a mouse treated with a combined treatment of nicotinamide and pyruvate. This is from a flat mount, and we do 3D reconstruction, so we can assess this around the entire eye. We do the same for the trabecular meshwork, and look how different this untreated trabecular meshwork looks to this treated trabecular meshwork. So these images are striking, and quantification of this is also showing we're getting significant differences. So we're correcting developmental malformations of the drainage tissues and IOP, so this could be important in childhood glaucomas. If these mechanisms carry over to adult glaucomas, which preliminary evidence suggests they will, this could also be important for lessening trabecular meshwork or Schlemm's canal death with age or disease and lessening the IOP elevation in those. I'm looking at this as a lessening of demise rather than a direct IOP lowering. Though nicotinamide may also have effects on directly lowering IOP in some contexts. If we look at clinical disease in these animals, anterior chamber deepening is a good measure of IOP because it reads out the total eye exposure in a mouse in a crude way. If you only do daytime IOPs, you might miss nighttime high IOP, but the anterior chamber of mice is pliable, so they have nighttime IOP, the chamber still deepens. So seeing a correction of anterior chamber deepening is very important and supports and extends the IOP data. In addition to the trabecular meshwork and cornea, we also improve iris adhesions and iris abnormalities in these mice. So we think this is very exciting data for childhood glaucomas that we hope is going to extend into adult glaucomas too, and our preliminary data are supporting that. So to summarize, age-dependent decreases of NAD increase the likelihood of metabolic decline and contribute to glaucomatous neurodegeneration. We've shown that nicotinamide treatment is potently protective and at a dose that's considered safe for most patients. Disturbed glycolysis and pyruvate metabol contribute to glaucoma progression, and pyruvate administration is also protective. Nevertheless, to keep doses low and for better efficacy, we're suggesting combinatorial treatments, and we're exploring this further in my lab and testing various analogs and trying to take this and identify other molecules which may synergize them to get a cocktail of hopefully safe molecules that might be protective from these nasty diseases and possibly even AMD and other diseases. The metabolism preserving treatments are highly robust in these mice in our lab and others. I stopped counting at 650 when we published results of 650 D2 mice. We've published 3 other studies since, including the Peruvid studies. So these are robust and have been reproduced by others. Not all strains have the same degree of correction as DBA2J, but in the LMX1B model, we're also seeing correction. And in the LMX1B model, we've shown perturbed metabolism and cell stress contribute to the IOP elevation itself. So this suggests that these treatments in glaucoma will have multiple fronts of action, which makes them even more important. Not only can they lessen nerve damage without changing intraocular pressure, because in those DBA2J experiments I showed you, the lower dose was not changing eye pressure, so that was a direct neuroprotection, but they also can protect the drainage tissues, so they can have a double benefit. I suggest systemically they'll have a benefit throughout the body for vasculature and other places in the body, and generally for health. And so I think it's a very good opportunity here. So to end any decrease in treatments have potentially a lot of promise for glaucoma. This is looking relevant to human glaucoma because there's an emerging and growing literature supporting mitochondrial susceptibility and NAD metabolism in human glaucoma. There's an increased incidence of mitochondrial mutations in human glaucoma in blood cells, suggesting systemic susceptibility throughout the body. Glaucoma patients have lower levels of nicotinamide shown subsequent to our work, and lower levels of Peruvid associated with worse glaucoma. Our first stage human clinical trials were positive in showing neural recovery, but they were small sample size, and we're continuing those trials. In the interest of time, I'm not going to go through those trials, but they were published from our group and another group, and there are now trials going on around the world. And we're moving to our next stage trials, which is already underway, both in our lab, our groups at Columbia with Jack Shoffey, Jeff Liebman, and Gustavo Moraes and others, and around the world from other groups. So it's an exciting time for glaucoma. I'd like to thank various members of my lab. Many of them have been important in this work. And I'd like to thank former lab members who've been important in different aspects of this work, and Abe was on the call. For metabolomics and the neurobiology work, Pete Williams did a lot of this gene expression work, and Craig Whitlock helped us with the metabolomics. Krish Kazatil has been very important in our development of Schlemm's canal technologies, and in single cell seq, there's Jian Qian, Taibo Li, and Dan Stemer, and others. So thank you very much for your time, everyone.
All right. Do we have any questions from the Zoom? If not, I think we'll start with Dorota.
Hi, Dr. John. It was really beautiful and very promising, especially on the treatment that is already ongoing. So I have a question about this, actually, first part. Your group 4 shows really a lot of changes in expression, but at this point, there is very few ganglion cells, right? And although I understand that your treatment with ganglion cells overexpression is working, do you think it's actually the real mechanism? Maybe it's actually the environment rather than intrinsic to ganglion cells. And of course, oral gavage will not distinguish.
No, I believe it's actually diet, it's not gavage, they're eating and drinking the food. At stage 4, there is no detectable ganglion cell loss. That's prior to neurodegeneration. You've got to get to later stages to get the degeneration. So stage 4 are still eyes prior to detectable degeneration.
Okay, that's ensuring. Thank you.
Thank you so much for that really interesting talk. So I was kind of curious, are there like intrinsic differences in NAD ratios or metabolism between different neuronal retinal cell types? And is this overall shift in like NAD to NADH ratios kind of dictated or determined by a single cell type, or is it more of like an overall metabolic shift in the retina?
Yeah. I'm not up with maybe everyone's studies recently, but from our studies, we can't distinguish that with the resolution of studies we've done at this point. Really, to look at that very accurately, I think you need the lifetime imaging methods like FLIM, etc. And we haven't had access to those technologies. So from our studies, what I can tell you, if you take a whole retina, you can detect these changes. Which cell types are contributing primarily to that, we can't tell you. But when we look within retinal ganglion cells, the genomic changes within a retinal ganglion cell itself is supporting those changes directly within retinal ganglion cells. The gene therapy also supports that. So we know ganglion cells are part of it for sure. What I can't tell you is what are the roles of other cell types, how are the cell types modulating this, and that's going to take a lot more work for the future to understand. And that's why it's particularly pleasing that the treatment's working if you give them systemically. For the treatment perspective, it's effective, even though we don't fully understand all the mechanisms yet.
Thank you.
Let's go to Dr. Nesburn from Zoom.
Thank you. Very impressive presentation, Dr. Simon, and very impressive data. I am interested to know how you're going forward with developing a clinical product. Is there a company that's formed, and are you teaming with Big Pharma in some way to bring this to?
Yeah, it's complex and it's been frustrating. I'm not at liberty to tell you everything, but basically I wanted to get things out there as quickly as we can to help patients. One of the troubles with Big Pharma is they see this as readily available molecules. And so they're very leery of it. So the nutraceutical approach, we're talking about forming companies. Some people have formed companies. We have patent protection to try to make this into a real treatment, hoping pharma would take it up. But none of the big pharmas have bitten yet. I'm surprised at that, but I think it's because they're these vitamin supplements and they're concerned about the doses of them. As we do more work and get these combinations and more tailored things, I think that will change. But one of my concerns right now is many patients putting themselves on this. I know the adverse events are there, but they take too high a dose or they're susceptible, that can be dangerous. So they need clinical monitoring. Many patients are going without that. Thankfully, it's relatively safe. But there's also the issue of when you buy things in a vitamin store, you don't know how much active product it is. You're concerned about contaminants. So what I would very much like to do is get a certified pedigreed product so we know it's high quality and good. And then people could buy that. So hopefully we can make more progress there and get it going. Sadly, it's been stuck for some legal reasons between institutions, where I changed institution and things, and hopefully we can get it unstuck soon and make some progress there. But there's other people trying to do these things as well. But we do have the IP protection, which will hopefully help it go to someone who's really got the funds to do it, a pharma, ultimately, once we find the best cocktail.
Well, it certainly looks very promising and would have to undergo, important clinical trials. So very exciting.
Yes, I agree. Thank you.
Hi, Simon. This is Gulab. Very interesting talk. I just had a few questions. In your DBA2J model, maybe I missed it, but have you looked to give the NAM treatment after they develop RGC pathology? So that was one question.
So we've not come back and done that extensively. And when the ages we've done different ages, but we haven't started it very late in the disease. What we have started is at ages where they've had ongoing high intraocular pressure, which is a great opportunity in the human population because people have high IOP for an extended period of time and they haven't yet developed glaucoma. So I've been more interested in heading off the glaucoma. In terms of existing pathology, the clinical trial is the best evidence of that, though we don't have histology. But in the clinical trial, what we actually showed was improving numbers of test points in the retina. Not all test points improved, and that makes sense. The worst test points, the cell's already dead, you can't bring them back. You're not going to generate a cell using this. But the cells that are under stress and non-functional, we restored function and improved visual function. Those small numbers, though needing confirmation, 2 different groups showed this in different studies in different parts of the world. So it's very promising. So I'd suggest, yes, you can stop ongoing disease, but you aren't going to bring a dead cell back. And so my interest in using this, I think its biggest advantage is it might go beyond the glaucoma clinic. It might become a general prophylactic for the population. If you can take a safe molecule safely, and much of the population takes it, like folate for neural tube defects, I think this is going way beyond conventional treatments and beyond dietary and getting away from eye drops and the difficulties. I think there's immense potential. I don't want to be naive and say this is going to solve everything and do everything. There's a lot more work needed, but I do see it as augmenting current therapies via different mechanisms. So I haven't had the interest in going when it's already massive cell loss, because I'm more interested in helping more people and slowing it down. And our evidence in the clinical trial is suggesting we're slowing it down when there's already visual loss. So I think that's the best way I can answer your question.
Yeah. So to follow up on that, your later study shows that this therapy could affect also the IOP. So how much is contributed due to lowering IOP versus actually directly protecting the RGCs?
That depends on the study. In the DBA2J model, our low dose had 0 effect on IOP. So in that model, the etiology of the IOP elevation, the doses we use and the timing, we did not modulate IOP. So that was a neuroprotective effect, independent of IOP. In this other model, on a different genetic context with a different etiology, we're certainly affecting IOP. And so in this model, I suspect we'll probably end up having a dual effect, an effect to IOP lowering and probably a neural effect in addition, though that can be hard to pull out in a model which is both going on at the same time.
Thank you. And the last question is during your initial presentation, you mentioned that you are looking into single cell RNA-seq on the TM, and you've already published the SC cells, and you're using the viral vectors. Are you using specific promoters for the TM cells, or because a lot of viral vectors also tend to transduce a lot of other cells?
And I can't tell you all of the details right now, it's too complex, but we've developed methods of delivering the viruses. We've developed viruses with certain surface receptors and proteins, and we get specificity with our methods. So we've got viruses for mice, where we can specifically transduce Schlemms or TM within reason, pretty largely one or the other tissue. And we're working on viral evolution studies. We don't have all of this done, where we're going to be isolating new types of viruses, hopefully the subtypes I mentioned, with the different trabecular measure and Schlemms canal subtypes. We want to be able to get them all specifically, hopefully. I don't know if we can achieve that, because the cell types, have similarities as well. So we're going to be taking both random mutagenesis and rational approaches to address this, and that's a big effort for the future. And hopefully we've got ideas how we can take this to primate and human, but our work currently has been in mouse.
Hello, Dr. John. This is Cezary Rydz from Dr. Skowrońska Krawczyk Lab. My question is about the NMN supplementation. So it's known that the photoreceptor function goes down with age. Have you looked whether the NMN supplementation prevents the age-related rod and cone functional loss in your mice? Thank you.
We haven't looked at photoreceptors in our mice. In our mice in the ages we're studying, the photoreceptors don't degenerate. In our first study on DBA2 mice, we take them very old, and in some environments, you can sometimes in some mice see some dropout there, but most mice it's not. It's highly specific to the ganglion cells. So we simply haven't done those studies. Other groups in models have worked on some of these molecules for the retina. I'm blanking on the name. There was a famous lab doing it, and they've even looked at a mutant gene that affects NAD metabolism and shown it protects in that, and they claim age setting as well. So there's evidence it could work in other diseases and in the photoreceptors as well. And given the growing field of metabolic influences in AMD, that's one of the reasons why we're very interested in testing these in AMD as well. But I can't give you more direct evidence. I'll just correct you on one point. We have not been using NMN. And we did not use NMN on purpose because it's a little murky. There's a lot of groups especially David Sinclair in Boston and others promoting NMN, and they've got their reasons. Not everyone agrees with all of the reasons. Some of these things are controversial in the field. So they like NMN. My concern with NMN was NMN can also contribute to axonal degeneration in some settings. And so I was concerned if we had excess NMN and it wasn't immediately being converted through and balanced in the enzymes, maybe it could actually be damaging. So I know it sounds odd, but that's why we specifically wanted to use nicotinamide, not NMN. But other studies are having good results with NMN. And so that might be one of the molecules which might prove in the future. Other groups are also using nicotinamide riboside, a different version of vitamin B3. They claim higher bioavailability. I don't know if I believe that's true for all of the data. You can interpret the data sets in different ways from the ones I've seen. But it might be a better molecule. We don't know. We haven't tested that yet. But there's also reasons I could argue nicotinamide is better. So the variation of which molecules to use, there's people in different camps. I tend to be open-minded, but for glaucoma, we've certainly shown nicotinamide works very well, and we might test some of the others if we are funding in the future. But I think for now, we're sticking with the nicotinamide.
Hi, Dr. John. This is Philip Kaiser. Really nice talk. I might have missed it. Did you test whether the nicotinamide treatment augments medications that are currently standard of care in your animal models, like a carbonic anhydrase inhibitor or latanoprost, that sort of a treatment?
Yeah, we've considered doing that, but so much time and money to do it all, we've decided against it. We're going for other types of studies first. We may try some of that at some point, or I'm sure other people are going to. Because the mechanisms are likely distinct, there's a very good chance they're going to help each other as long as there's not some adverse interaction between the chemistries or signaling pathways in some way. But we haven't actually done that. I think that's work for the future. There's some we've considered doing, such as the latanoprost and some of the common current glaucoma medications, but we actually haven't done it. The model we're working with also has a very severe and florid pigment dispersion. And some people have reported protections with some of these molecules or damage in the ciliary body. But it's a severe model, so it's not easy. And that's one of the reasons why our results are so encouraging.
Right. I was just asking, I would imagine clinically that might be a good application for this, somebody who maybe has sort of a suboptimal response that you wanted to, try to add that on to achieve, the therapeutic goal. And then another thing to consider is that might actually allow you to get the dose of nicotinamide down again, because you have this sort of potential synergistic therapy between those things. But just
yeah, I absolutely agree. I'm thinking of it that way. I think there's multiple uses here. One is you might be able to treat more people and stop IOP elevation in the first place, and they don't get to need the other drugs. The other one is augmenting the other drugs and possibly decreasing the dose of the other drug or the nicotinamide in different ways. And I think there's so much complexity and so many possibilities. There's a field to us to work a lot of these things out, that I think they're very exciting possibilities.
Wonderful. Thank you.
I guess I have a question. Do you think that your nicotinamide treatment could be extended to patients with normotensive glaucoma?
Absolutely. If I look at normotensive glaucoma, it's probably, though the evidence isn't fully in and there's a lot of work needed, but some people have suggested vascular abnormalities and other issues. And it just might be, the proportion of what they've got that's vascular or other features is more important for their disease. Being as nicotinamide has effects on blood flow, it can promote blood flow. It can promote the health of endothelium in some studies, though there's the caution from the recent Hazen paper I mentioned. But it's quite possible it'll help in normal tension glaucomas as well. One of the reasons I'm enamored by this is I see it as possibly, if you're just increasing the health of cells and you're allowing cells to fend off aging and fend off a variety of insults by giving them more energetic resources, more antioxidant resources, they can build these resources up with a more NAD, more glutathione, etc. They should be able to fend off many stresses better if they're better equipped, if they have the resources to do it. And so I think, they could have broad applications across different initiating etiologies or importance of different etiologies. So I think it's well worth testing.
All right. Are there any more questions? If not, let's thank Dr. Simon John one more time.
Thank you, everyone. Good to see you, Chris.
John, thank you so much. That was fantastic. And I think what you said about the companies not picking it up, and particularly once you get to the multiple active ingredients, that is going to be absolutely no-go for the companies because of the clinical trials cost and number of patients. But
Yeah.
I think like you have this case of vitamin A and other vitamin supplementation for AMD that was carried by National Institute. Maybe this really approach, as well as we were developing a very, very effective treatment using systems pharmacology, where you using 3 different drugs, the combination of well-tolerated, well-known drugs and well-known pathways, that this maybe belongs to an institute to carry this kind of work where the financial upside is hopefully not important, but that will help patients. We will have coming up Nature Communication paper on dogs and mice and many different models that our combination works also so well. But this money-driven approach to having something that is patentable and based on a molecular entity approach, it doesn't benefit patients that much.
Yes, I think you're right. The NEI is a valid approach for the future. Let's hope funding and things can improve, and with the economy and other things, and that there'll be a healthy institute and it can keep pushing these studies forward.
Yeah, this again, chronic diseases will require a different approach. It's not going to be a bullet, one type of drug affecting all of the pathways and so on. That is not going to be likely happening. So hopefully we can get together and talk more about this, because it's just several very good ideas that will not find a commercial interest, and but they are still very effective treatments.
Exactly. Yes, we should, Chris. That would be great.